gene of interest using PCR. Amplification means multiplication of gene of interest, the gene from which either plant or animal which is interested, that gene is multiplied using the technique polymerase chain reaction. Polymerase chain reaction. Okay, so this technique is introduced by the scientist Kary Mullis in 1983. Kary Mullis in 1983. He was awarded Nobel Prize in Chemistry in 1993. So when we identified desired gene, so the desired gene can be multiplied through two processes. When we can identify the desired gene, so the gene, if you want to multiply, we can be multiplied through two methods. One is using cloning vector. Using cloning vector. One is cloning vector, another one is polymerase chain reaction. So cloning vector, it takes much time. But another quicker way and far more best technique, greater technique, within a short span, within few hours, we can be produced billions and trillions of copies, trillions of desired genes. So through this, it takes time, much time, but through this technique, so within few hours, we can produce billions and trillions of desired genes. That is called polymerase chain reaction test. When the DNA is not available much, a little fragment is available. Through that little fragment, we can be multiplied millions of copies. Okay, so that is what we will see. Okay, ma? That's all. Now, PCR technique involves in 3 steps. One is the PCR involves in 3 steps. The first step is denaturation. The second one is annealing of primers. And third step, primer extension. Through these steps, we can be finished PCR cycle. First of all, denaturation. The word itself saying the meaning denaturation. See, the desired DNA is taken into the test tube. In this one, desired DNA is there. The desired DNA is taken into the test tube. In this one, chemically synthesized chemically synthesized primer. So chemically synthesized primer. This is called primer. So 5' to 3'. Chemically synthesized primer, which contains 20 to 30 nucleotides. 20 to 30 nucleotides. Those 20 to 30 nucleotides also called oligonucleotides. See, already what is primer? I said on 6th chapter DNA replication. Before replication, the primer must be added. So that primer helps to replication of further DNA. That is already we have seen that. So where we have discussed, let me show that. So here, So before starting replication, here we needed to add primer. In textbook not there, I said. So with the help of this primer, DNA polymerase enzyme will be continue. Here also primer, RNA primer, RNA primer, RNA primer. I said already. Okay, so that primer helps further replication of DNA. Okay, so that primer here artificially chemically synthesized primer which contains 20 to 30 nucleotides. 20 to 30 nucleotides means what? What will be there? Adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine, like that. 20 to 30 nucleotides. So this the primer which contains 20 to 30 nucleotides, such type of primers are called oligonucleotides. 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 Okay, so the result DNA is added into the test tube. With it, 2-3 primers will be added. 2, 3 or 4 primers are added, and then it is heated to about 90 degrees centigrade. It is heated to about 98 degrees centigrade. When it is heated to 98 degrees centigrade, when it is heated to 98 degrees centigrade, then automatically the double-stranded DNA, 5 prime to 3 prime, 3 prime to 5 prime, 5 prime, double-stranded DNA. Here, double-stranded DNA. It's complementary adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, thymine, guanine, cytosine, thymine, adenine, like this way. Like this way. Okay, so this is called double-stranded DNA. The double-stranded DNA will be like this. Okay, double helical DNA or double-stranded DNA, 2 chains. So are taken to the test. When it is heated to 98 degrees centigrade, then this double-stranded DNA dissociates, separates into 2 strands. These 2 strands becomes 5 prime, 3 prime, 3 prime, 5 prime. This double-stranded DNA became single strand. Due to heat, this double-stranded DNA automatically split by breaking the hydrogen bonds present in between these 2 strands. Then it becomes single strand. Then it becomes single strand. Then it becomes single strand. That is called denaturation. The word only saying the meaning, denaturation. So when we will heat about 98 degrees centigrade by adding primers which contains 20 to 30 nucleotides, those primers are called oligonucleotides. Then double-stranded DNA becomes a single strand. Single strand. So the double-stranded DNA dissociates. Dissociates means separated. Dissociates into single strand. That is called denaturation. Next, editing of primer. Next, after separation of 2 strands, so this test tube is cooled. This test tube is cooled about 68 degrees centigrade. 60 degrees centigrade. It is cooled to 60 degrees centigrade. Okay, so when this DNA is cooled at about 60 degrees centigrade, then what happens? It's complementary reassociates. It's complementary reassociates by attaching primer. This is called primer. Okay, when it is cooled to 60 degrees centigrade, at that time, here the single strand automatically, according to the complementary, reassociates by joining primer. Here also, by joining primer. This side we will show. This is called So this is called annealing of primers. Annealing means joining of primer. Annealing, joining or sealing or ligase of primer. So when this primer. So for this stand this side, for this stand this side. Or else here also we can draw, no problem. Okay, so primer. So the primer is attached to the single strand according to the complementary base pairing. According to the complementary base pairing, the primer is attached to the single strand. That is called annealing. Annealing means joining of primer. Annealing means joining of primer. Annealing means joining of primer. So the primer is joined. Okay, so here for our diagram connectance I have drawn here. So here also you can draw. Okay, so here the primer is added. It is the primer. This is primer. The primer is added. Here the primer is added. So that is called annealing of primer. So the primer is added at one end of the another strand. One end of the other strand. The rest of the strand is left like that. So the rest of the strand slowly replicated. Okay. So that is annealing of primer. Next one is primer extension. Primer extension. So for joining primer with the one strand, here complementary. This complementary, it will be joined. According to this complementary, the primer is joined. According to this complementary, the primer is joined. So the primer is complementary to another strand. This primer is complementary to this strand. Here one heat stable variety of engine. Thermostable, heat stable or thermostable variety of enzyme is isolated from the bacteria, that is called thermos. Thermus aquaticus. Thermus aquaticus. From Thermus aquaticus bacteria, they will eliminate DNA polymerase. This enzyme is called polymerase. Thermus aquaticus polymerase. It is also called DNA polymerase. DNA polymerase enzyme is isolated from the thermostable bacteria, heat stable bacteria, that is called Thermus aquaticus. That means what? So even higher temperatures also this enzyme can be performed effectively. This enzyme works effectively, but some of the enzymes at higher temperatures becomes inactive. But the Thermus aquaticus, that's why it is called thermostable bacteria. At high temperatures also it can work effectively. Thermus aquaticus. Okay. So Taq polymerase. So this Taq polymerase is Thermus aquaticus polymerase. This is also called DNA polymerase. Enzyme is isolated from the Thermus aquaticus and added here. DNA polymerase added. Then this DNA polymerase continue its primer extension according to the complementary rule. Here also primer is added according to the complementary rule. According to the complementary rule. Here also same. According to the complementary rule. According to the complementary rule, DNA polymerase continues formation of complete nucleotide bases according to the complementarity and base pairing rule. So see here, one DNA became two DNA. One DNA became two DNA. So this is called polymerase chain reaction. Yes. Polymerase chain reaction. Yes. Okay. So in this one, Taq polymerase play a major role in the extension of primer. Okay, that's why this is primer extension. Primer extension takes place. The primer extension takes place with the help of DNA polymerase. DNA polymerase engine play a major role in the extension of primer according to the complementarity and base pairing rule. This process is called polymerase chain reaction test. Okay, that's all. Here, multiple copies are amplification. So multiple copies nothing but amplification. See, through these three steps, we can produce the multiple copies. 2 DNA becomes 4, 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, and so on. In such a way, multiple copies are formed. The steps above mentioned, that is denaturation, annealing, and primer extension. For these three steps, the time requirement is 2 minutes. 1, 2, 3 steps mentioned above. It will finish within 2 minutes. The requirement of the time for these three steps is 2 minutes. Within 2 minutes, the heat and cool process is finished. So the time taking for heat and cool is 2 minutes. Within 2 minutes, one DNA becomes 2. Within 2 minutes. Within 2 minutes, 2 DNA. 4 minutes, 4 minutes, 8 DNA. In such a way, the multiple copies of the DNA are formed. Here, the enzyme DNA polymerase. The enzyme DNA polymerase is a heat stable variety of enzyme. No need to add a number of polymerase enzymes. One enzyme is sufficient to produce number of copies. Okay, even it can stable at high temperatures also. Okay, so since DNA copies are doubles for every 2 minutes, within 20 minutes, in about 20 cycles, in about 20 cycles, that is 40 minutes, the DNA copies becomes millions. In such a way, 20 cycles means 40 minutes. 22 to 40 minutes. Within 40 minutes, the DNA copies becomes millions. In such a way, few hours, we can reproduce billions of DNA copies. Those are all the DNAs are desired DNA. Those desired DNA, we can be transferred into the plants or animals, etc. Okay, so nowadays PCR technique is very famous than cloning process. So some of the places cloning are using PCR technique, are using both are using. Okay, so this is the explanation about PCR. So in textbook, one diagram has given that. Okay, so that diagram is very important. You have to practice that diagram also. In the text book, one diagram given that. The diagram is important for 5 marks. So simple, this is only, so proper way you have to draw. Okay, so you have to practice the diagram. It must and should come for 5 marks. And some conditions of these questions, this question they will ask for about 5 marks. Okay, so within 2 minutes, the cycle is completed. That is called PCR technique. Next topic, already we know that. So insertion of recombinant DNA. Into the host cell or organism. Insertion of recombinant DNA into the host or organism. Already we have discussed very much. Okay, about insertion of recombinant DNA into the host cells. First, so when this is vector, This is called vector. This vector is transferred. This vector is combined with the foreign DNA. This vector is combined with the foreign DNA. Like this, according to the complementary. So this is vector, this is foreign DNA. This is foreign DNA. So the foreign DNA is combined, ligated or inserted at the vector. Then it is called recombinant DNA. So this recombinant DNA is transferred into the E. coli bacteria. This is transferred into the E. coli, Escherichia coli bacteria. Okay, so there are a number of methods. So indirect method, direct method already we have seen. Okay, so directly recombinant DNA does not accepted by the bacteria. First, the bacteria must first be ready to take up the foreign DNA or recombinant DNA. Then only further process is continued. So that is already we have seen. For transferring recombinant DNA to the bacteria first we have to make it ready. We have to make it competent to take up the DNA. So to make it bacteria ready, already some steps we have discussed. Okay, that is indirect method or direct method. Okay, so this bacteria have to treat it with electroporation, incubation, etc. techniques we have to use. So with the help of that, so the cell membrane is ready to take up the recombinant DNA. So that is already we have seen here. Okay, so that's next here. The recombinant DNA bearing gene for resistance to antibody. The recombinant DNA. This is called recombinant DNA. This recombinant DNA bearing gene for resistance to antibody. Listen carefully. This is also already I said that. So when we will insert foreign DNA, here tetracycline lost the tetracycline gene lost resistance power. But still ampicillin resistant because ampicillin will be there. Ampicillin resistance. Here tetracycline resistance. So the recombinant DNA bearing gene is recombinant DNA containing gene for what? Resistance to antibiotics. Resistance to antibiotics. So here for example, so the recombinant DNA containing ampicillin resistance power. So this ampicillin containing gene, if it transferred into the E. coli, ampicillin containing gene, if it will transfer into the E. coli, it means E. coli, Escherichia coli cells become resistant to ampicillin. Because here ampicillin gene does not disturb. That's why this recombinant gene containing ampicillin resistance. Ampicillin containing recombinant DNA are transferred into the E. coli cells. This is transferred into the E. coli cells. Okay, ampicillin containing gene, if it transferred into the E. coli cells, it means this E. coli bacteria becomes resistant to ampicillin. This E. coli bacteria becomes resistant to ampicillin because ampicillin gene is intact, but tetracycline gene is not intact. So due to the insertion of foreign gene, tetracycline gene lost resistance. Okay, tetracycline one gene is lost resistance to tetracycline, but still ampicillin resistance. Okay, that is knowing point only here. Once again, I am repeating the point. Okay, see here, recombinant DNA bearing gene for resistance to antibiotics. That is especially for ampicillin. For example, ampicillin containing recombinant DNA, ampicillin, this is DNA containing ampicillin resistance. Ampicillin containing DNA, if we will transfer into the E. coli, that means the E. coli cells becomes resistant to ampicillin. So this recombinant DNA is called transformant DNA. So this ampicillin containing transformant DNA or recombinant DNA, when we will transfer into the E. coli, this E. coli becomes resistant to ampicillin. Okay, that is one statement. This is one statement, it is one statement. Third statement. Now, if we transform, if we spread this recombinant DNA on agar plate containing ampicillin. See, now, if we spread this recombinant DNA or transformant DNA on agar plate, see here, this is agar plate, here agar plate. So, in this agar plate containing ampicillin resistant gene. So, now, if we spread this recombinant DNA on agar plate containing ampicillin, the ampicillin containing gene will grow. Ampicillin containing gene will grow. Others will perish. Perish means die. So only when this recombinant DNA is transferred into the agar plate containing ampicillin, only ampicillin containing gene will grow on the agar plate. The gene which containing ampicillin resistance, that will grow. Others will not grow. That means what? Recombinant DNA will grow on ampicillin containing medium. Non-recombinants will not grow. They will die. Only the gene, the recombinant DNA which contains resistance to antibiotic, that only will grow on agar containing ampicillin. Others will perish. Perish means will not grow. They will die. Okay, this is one statement. Fourth statement. So in this fourth statement, one is able to antibiotic gene able to select recombinants. One gene is able to select recombinants, that is ampicillin resistant gene. Ampicillin resistant gene is able to select recombinants due to the presence of ampicillin resistance protein, due to the presence of ampicillin containing gene. So that ampicillin containing gene in this case acts as selectable markers. This is the point on that only I said that ampicillin and tetracycline both will act as a selectable markers. In these two, one will select recombinants, another gene lost resistance due to the insertion of foreign gene. Due to the insertion of foreign gene or alien piece of DNA. So in such cases, ampicillin resistance gene acts as a selectable markers. So this gene selects transformants. Transformants nothing but recombinants. Ampicillin selects recombinants. Tetracycline gene, it gets deactivated due to the insertion of foreign gene. So why tetracycline gene gets inactivated here? Its gene is cut off, so that the complete structure became incomplete. But the ampicillin resistant gene is not cut. So that the ampicillin resistance gene selects selectable markers. Ampicillin resistant gene acts as a selectable marker. So with this insertion of recombinant DNA into the host cell or organisms. Okay, so with this all steps are finished in the process of recombinant DNA technology. Next, final one topic is there: obtaining foreign gene product. Obtaining foreign gene product and downstream processing. After obtaining the products, what are the techniques we have to follow? That is called downstream processing technique. That is what we will see tomorrow. So today meeting session is finished. So get ready for another class.